Hello and welcome to Food for You and the Planet, a fresh new podcast that explores healthy and environmentally sustainable food solutions. This podcast is brought to you by Healthy Plan Eat online farmers market. I'm Dr. Rosemary Ostfeld and I'm your host. Together, we'll hear from leaders in environmental science, agriculture, medicine, nutrition, entrepreneurship, and more. Be sure to hit the subscribe button to hear all the latest in healthy and sustainable food with Food for You and the Planet. Today's episode features Dr. Michael Mazourek, Associate Professor in the School of Integrative Plant Science, Plant Breeding and Genetics, and Horticulture. Michael Mazourek is the Calvin Knoyes Keeney Associate Professor of Vegetable Breeding at Cornell University. Michael is a breeder of peas, beans, squash, cucumbers, and peppers, and has released numerous cultivars and breeding materials that are shared by small regional seed companies and incorporated into breeding programs of the world's largest seed companies. Michael's specialty is biochemical genetics in vegetables. He explores the diverse phytochemistry that plants use to repel pests and herbivores, reward and nourish pollinators and seed dispersers, and cope with environmental stresses, with a goal of harnessing to maximize nutrition and sustainability. Michael shares the craft of plant breeding with students at Cornell through grower conferences and field days. Thank you so much for being here today, Michael.
Thank you for the invitation.
So can you give me a short overview of your plant science research at Cornell University?
Sure. So it's shifted a little bit over the years. So a lot of people know me as the miniature butternut squash guy. That was a great honor. We continued a legacy of efforts of squash and the honey nut squash breeding program at Cornell and a lot of other vegetables I steward. So I am working on trying to create, I think, for myself, kind of the ultimate little set of veggies I would want to grow in my garden. We have a big emphasis on how to help local regional farms be competitive with distinctive produce that stands out with better quality, better flavor, or even just help the farmers with the traits they need to be able to get through the season and have a good harvest, like disease resistance. So we are now doing a lot with looking at the nutritional content of fruits and veggies. And it's pretty high to begin with, but there's some things we can really do to make things shine. Like peppers are a great source of vitamin C, all the eye health pigments. And so, whatever we can do to help people consume more is going to help their health. So, we're creating those cultivars. We're also diving in to see what's the science behind it. So, we are doing some big data models with doing the whole genealogy, all the gene sequencing of the varieties we're creating, and also zeroing in on how do certain genes tick, what makes them work, and we have some great collaborators we work with on those.
How did you become interested in plant science, food, and agriculture?
It was to the credit of those around me, for sure. I I grew up on a local farm here in the Finger Lakes, down the road from one. My dad's a dairy farmer, equipment dealer, makes livestock feed for local farmers, fixes everybody's everything. And I found myself going to school for biochemistry and looking at where all the compounds we can take. When our health goes awry, what are the compounds we can take? There are interventions that can like help offset whatever is going wrong in our bodies. And I discovered late in my undergraduate career, so I graduated and was totally lost when I discovered that plants are making similar compounds every day. There's no one in the back room of a pharmaceutical company dreaming up these new compounds. We're finding them in plants and microbes and then picking one compound out of a family of related compounds that they can be scaled up and put into pills. So I became very interested in these compounds in plants, and like every flavor we're after, like any spice in your spice cabinet is making a set of aroma compounds and flavor compounds that delight us, but they're actually intended by the plant to help direct the other organisms in its environment. So they'll be to deter feeding, like the caffeine we have in our coffee. That's not so we'll grow more coffee beans and drink more coffee. It's to defend the plant from insects. And so there's compounds that interact with our physiology in productive ways, counterproductive ways, and for the plant it has its own agenda. So I became really interested in that, and then ended up rescued from unemployment by my advisor, the awesome Molly John. So I was totally lost, and I started working with her, and I discovered plant breeding is a way to help get these compounds um, into people's diets, and also, for the local farmers that are important to me, a way to also give back to them. So it just came together. Um, I'm so lucky.
So, what is the history of plant breeding? How and why did it begin?
So for the true history, we have to go back thousands of years. And, imagine there's no gardens, no grocery stores, no crops. So it's the intrepid souls that are tasting plants in the wild. Sometimes people are getting sick and dying from this, but it's hard times. And so people are first domesticating plants from the wild. Um, and it's a matter of finding something that's edible, saving seeds, kind of replant the ones you liked. And then over time, you're in a position to identify, well, here's a new one that just kind of showed up from all the outcrossing and selection that's been going on. And so, for thousands of years, the origin of agriculture is people domesticating plants from the wild. It's evolved quite a lot since then. We now know DNA and genes, and we can design different cross pollination schemes. And depending on how complex the inheritance of a trait, I can tell you how many plants to grow and what different pollination scheme you might want. And we can sequence their genomes and understand exactly what's going on. In some cases, a lot of it's still yet to be discovered. But so, yeah, it owes its origins to the first intrepid souls that were finding plants in the wild, finding plants in new locations, and finding what was good to eat and making it better.
Why is plant breeding important?
Right. So plant breeding, I like because it is probably the most powerful biological technology we have. Right? If you think about every other technology in the world, how we've harnessed the electricity, and what we're doing now on Zoom, there's so many technologies that we've developed in all these different areas of our lives. So plant breeding is harnessing evolution. And so all around us, our preferences are changing. We have new growing systems. People want to grow in high tunnels or greenhouses, or there's new pests. They're evolving. Insects and diseases are moving in global trade. So we have to evolve our crops to keep up with the world around them, and make them better, and how we conceptualize better. So all of this, how do we address it? Well, the most effective way is plant breeding. So it's finding the crossing the best with the best to make something even better. If there's something you want in agriculture to be more reliable, to be use less resources, be more sustainable, to be tastier, sweeter, longer shelf life, to capture qualities of those heirloom crops and make them more practical to grow. All of that, if you want those changes, plant breeding is what does it.
What are some of the methods for breeding plants?
So the easiest, the most straightforward method, it'll look a lot like you might have had in high school, talking about Mendel. Um, so we call it the pedigree approach. And so, if you draw it out, it looks like a family genealogy. But you'll find two plants that complement each other in some way. Maybe there's a plant you really love, except it has a fault. Or there's two plants where you want something that combines the best of both in a more complicated way. So the simplest approach is if you cross pollinate those, you end up with an F1 hybrid. It's unstable. They don't breed true. We've all heard that. And so, but that's where the magic happens is when you save the seed from a hybrid, you get all the permutations of all the different characteristics of the parent. And then um, it's a matter of he's basically going pick of the litter through that until you find your favorite one and you save seed from that and just iteratively do that until you get something that's really stable, uniform as you need. Um, so a lot of the vegetable varieties that we enjoy now have been bred through that method. There's some other approaches where you might decide, well, you've crossed two things, but you really need to be more like one of the parents than the other. So in plants, then we would do a back cross. So we can keep crossing back to that parent and selecting for all the characters while adding more doses of what make that nice. There are some more complicated variations on this, and there's an alfalfa. The alfalfa breeding is very complicated, how you manage populations. Some crops need to exist or often exist as hybrids because as you self-pollinate them, they have inbreeding depression. So those plants you need to be breeding pairs of parents to bring together. But all of that tends to be looking at just single traits. So is kind of a good way to work with simple traits in general. Increasingly, we're moving past the Mendelian biology that really kind of looking for those couple magic genes that do things and really looking at what makes a cultivar that is perfect and does everything a grower needs. And maybe of growers in different states, they need that, but it's a little different. To have those finesse tweaks, you need to work with it more as a population. And so a lot of the breeding programs that are developing the some of the cultivars we're looking at that are really work at the real nuance of productivity and all those traits. They're working with large populations, and I was just doing a review session. So on the board for my class, there's a lot of it that if you look at a population and all the traits you look at, you're going to kind of get this bell curve distribution where a lot of the plants are average and some are below average, some are above average. And so we want to shift the mean over time. And so we want to shift that kind of that bell curve on the wall towards varieties where we can just year after year just steadily improve the really complicated traits, ones that are controlled by many genes, more than we can just, one or two that might give us different colors that make something spicy or not. So these processes use quantitative genetics, and increasingly for that, we are sequencing plants' genomes and using computer models we are training to be able to predict which plants would be make the best pairings to kind of continue to get that gain. So still embracing all the complexities of the whole plant genome and just really the full what evolution would be doing and just nudging that along a lot more productively.
How does the type of breeding you do differ from genetically modified organisms or GMOs or work done using CRISPR?
Yep. Yeah, so those techniques you mentioned, I'd put them all in that kind of transgenic recombinant DNA bucket. They're all techniques where there is one or several genes that you might want to go in and surgically alter with some Usually in tissue culture, you would use a set of molecular biology reagents to change a couple gene sequences. Maybe one, maybe a dozen. Then regenerate a plant from tissue culture, and it would have hopefully those changes you're after. Um, it works well if you want to turn a gene off. If you want to remove thorns from roses, if you find the rose gene, you can turn off the thorn gene and then thornless roses. If you want a tomato to be purple, you can put in the blueberry gene for purple and have as much anthocyanins as a blueberry. Well, I already have blueberries, but whatever. So it works well for these big changes that can be pretty simplistic in what they're doing. So what we're doing is we will sequence the genome, use information, landmarks from the genome to guide us so we kind of have a map to guide us in the process. But we're crossing plants that could have differences in most of their 30,000 different genes they have in them. All of the regions of the genome they're still learning that are important sequences, but not genes themselves that modify, attenuate the expression of those genes. And so, one approach that the editing or transgenic approach is if you discover what a gene is and does and all about it, then you can go in and surgically alter what form it is in a plant. You can maybe translate information from different kingdoms of life. In mine, I can work with all the information we have yet to discover in addition to what we already know. And if you want to have a plant that's super high yielding, resists stress, has all the exact shape and flavor and color, that's thousands of genes and genes yet to be discovered. So in my approach, we embrace the unknown and can push it forward anyway. They have some different strengths, but I like mine the best.
What are some of the types of plants you've bred?
That's a great question. I'm known for doing the work to finish the little mini butternut, honey nut, and all the other miniature butternuts out there trace themselves back to me or Cornell. My predecessors, Dick Robinson and Molly John, were two great plant breeders before me. I do a lot with cucumbers. There's a disease, downy mildew, that will wipe out all the cucumbers in the humid areas once it moves in. Takes a lot of pesticides for growers to control, and people like me would prefer to eat cucumbers without those pesticides on them altogether. Not to mention save the grower a bunch of money. So using some resources left to me by the other great plant breeder Henry Munger, who are able to quickly deliver those solutions to growers. Um, I really like playing with my foods, so we also have habanada peppers, the no heat habaneros. We work on rainbows of these flat snap or sugar snap peas. So getting playing with the biochemical genetics where we can boost color and flavor and look how they interact and do some science and create some tasty new seeds.
What are some ways plants can be bred for great flavor? And can you give some examples of some varieties you've created?
Sure. So probably refer back to that list a little bit. So I think the key to breeding for flavor and what we do that's a little bit different than most is for if you're going to create, say, a great tomato, the easiest step is to walk down rows of thousands of tomato plants looking to see where do you see a healthy plant that's loaded with perfect red orbs. You can do that really quickly at a walk and you can see which ones stand out. It becomes a totally different uh, project then if you want to breed for flavor because you have to taste each and every one. And that is fun for the first hour. and then becomes this horrible sentence thereafter. When we were working with watermelons, after you have tasted hundreds of watermelons, your body becomes so used to all that sugar on the liquid that as you become thirsty and you just go to have like some regular plain water, that water tastes so harsh and abrasive because you're just used to like syrup. It's hard. With habanada, so it's a no heat habanero, we had a combination where we used some tools to help us, but then had to rely on tasting lots and lots of peppers. So we knew the gene that made the peppers not hot. And so that's critical because if you want a not hot pepper, you can only eat so many habaneros in a row until you can't taste anything anymore. So we are able to look through the flat of seedlings and find pretty accurately, not perfectly, all like at the ceilings, which ones had the genes for being hot or not. So the same way you might get a test done at the doctor's office, you can also get a little test on the plant's DNA. And so we only planted the ones that had naturally inherited the genes to not be hot. A couple of errors, you find those as you bite into a habanero. You're very aware of the error rate. There's a couple, but a couple's enough. Um, and then, as you have these not hot peppers, um, you have a range of flavors. Some have strong flavors. Sometimes they're good. Sometimes they're bad. Sometimes, they're very soapy. And then you have ones with really mild flavors. So then it's a process of we taste each pepper off every plant a couple of times. So it's probably a few thousand different tastes we would do every season to be able to select the ones that have no heat, look good, and also taste really good the way we wanted. Um, we in other cases, I mean, it typically just involves a lot of tasting, but I love it when I can get input from someone as to a cool flavor they love in a crop that I now can increase what I'm looking for as I'm out there doing the tasting. Um, but the great input from chefs we've had in terms of what they want to do with a crop or how they work with it. And so as you look at how people pickle crops, so that makes it trickier for us, or roast it, then we have to harvest everything, bring it in. But so a lot of these cooking techniques will really change the flavor or change what you're looking for. So with beans, we were able to figure out we have these little half pint, maybe they're half cup, these little mason jars. And we put a few beans with water in them, put the tops on lightly, and put sheet pans of them in ovens. And then we cook them all. Sometimes they get a little over or undercooked. We don't hold that against them. But you can taste many beans after they're cooked, like dry beans, and see how they hold their color or not. And so, yeah, it's there's some tools we can use. I can tell you more about those, but it's mostly eating and knowing what you like about a crop is the key.
Sounds like a lot of fun taste tests.
Yeah, that's true, but you need to kiss a lot of frogs to find your prince.
How have you worked with local growers to breed plants?
Yeah, so I think that's been really important to be able to make sure you're satisfying what they need and what they're trying to do and how they're working with the crop. I think the helping the local growers is important. Beyond, they are my community, and so it's important to give back, and that's why we have these land grant universities in all the states is for that. Um, but also here in New York, there's some crops that there's not much work done otherwise. You see a lot of work done in Florida and California because that's the big markets. So most people tend to focus on that. But those crops don't work as well here. The same reason you don't see maple trees in Florida and you don't see palm trees in New York. So there's some differences. Um, So I love to be able to get seeds out onto a grower's farm, uh, and they can help tell me if it fits what they're looking for. The chance to be able to walk a field with a grower is fantastic. You get to kind of see what they see. Are doing now a lot of work with Norwich Meadows Farm. So as we are doing some testing there, and I have the chance to just help the harvest crew, I learn a lot. So they showed me as they're picking cucumbers, you can get a cucumber with a long stem, then they're able to, with one hand, kind of pinch it with their thumb and kind of snap it off. And then they can pick with both hands. So I now know how to make them what they're after. I had the good fortune to work with Blue Heron Farm a few years ago on of creating a lot of new vegetable varieties. It's kind of a key testing zone. And so some of the input, I was told the things that were interesting, but not a good fit for the farm. Got some really good insight as to good resistance on cucumbers, but the cucumbers need to be a different way. These little butternuts were intriguing, but get rid of these light bulb shapes was a quote I remembered. And, we knew we had a hit when it takes some back and forth. When we thought we had a good squash and the farm didn't quite agree, but after a little bit of back and forth, Robbins Kogi nut was selected right there at Blue Heron Farm, and they really helped us to see that not only did we like it, it would work for them, and that was such great insight.
How can plants be bred to be more disease resistant?
Yeah, so disease resistance, you can take a couple different paths. The easiest path we always hope for is that there's one single gene somewhere that's going to make a plant resistant. We can have a DNA tool then to look for it and be able to find the plants that have that and skip working with the actual putting the plant out and inoculating it with the disease. The um, other approach is a little bit more laborious, but it works really well. So we can often we're going to put plants out in the field where the disease is, and actually kind of in that setting discover which plants are the most resistant. So for our downy mildew resistant cucumbers, um, it's really hard to work with cucumbers. They grow really fast, they grow together. You have to pick a lot of them to keep them going in the field to do those assays. We have an approach where we plant the cucumbers out in the field. The disease has its way with them. We find plants that are still alive, still have cucumbers on them. Uh, and then we can take cuttings from those, bring them back into the greenhouse, and then it's really a chance to intermate the survival of the fittest. So it's really great to have the actual disease there. If there's shortcuts, that's great. There's a problem with that downy mildew resistance breeding is unless we have tools to predict which plants carry the genes for resistance, we're relying on our one shot a year to work with that disease. We don't want to introduce it at some time where it's going to get out to the local farm, so we wait until it shows up and finds us. So unfortunately, that gives us one shot a year. It'd be nice to be able to be a little bit more productive the rest of the year.
How can plants be bred to be more resistant to extreme weather events such as droughts or floods?
That's a tricky one because you never know which one you're going to get. Um, so we have the most straightforward way is to go ahead and just go put the plants in that environment. And so at Cornell, we use our high tunnels, so it's just a thin sheet of plastic over a metal frame. And we use that to exclude rain. And so we can simulate drought pretty well. And so then that's really helpful for being able to see how the plants perform there. Sometimes we can find collaborators in different parts of the country, in the desert that just have reliable drought. Um, In doing that, though, it's not just as simple as rain is the only difference. So, Arizona, you can get sunburned on Christmas Eve there, which is very different than what happens in New York. So, there's a lot of differences. So, just the chance to recreate that environment you're looking for and to be able to find plants that are available through our government has a national germplasm system in the US. It's a fantastic collection of the diversity from around the globe. So we often can make a lot of progress by screening that, tapping into it. But also there's some land races and other materials growers might have that might be a hot tip of something to start with that's even better.
What do you see as the future for organic, open pollinated plant breeding?
So that one is complicated. My crystal ball is a little needs more Windex to do a great job on that one. But I think there's a lot of dimensions to that. I think one of the important things to embrace is a big part of it is the kind of a little bit more of the usually boring behind the scenes stuff. Though there's a lot of stuff in terms of the consumer facing things, the grower facing things about like the flavor and the new things we're very excited about. To do that, I think it really relies on a lot of there are issues with access to the seeds and germplasm these days. So increasingly, there's a lot of patents that companies take around the use of a variety. So there might be a trait, and so you can Google patents about having better flavor in a crop. And so as these patents come out, someone will be not only tying up the use of a variety that's very flavorful in plant breeding by others, but even that path they had and the types of flavors, and they owned that whole space for 2 decades, which is a long time if you're looking for innovation and tying stuff up for 2 decades, and where no one else can use the either those seeds or the concept is something that I hope the organic community can do more to rally around. I think there's things you'll see in some of the codifying language for organic about how patents can be an obstacle to being able to continue to make the advances and innovations I think we need. So hopefully organic can do more to get behind that. It's also in having open pollinated seeds, there's still some need to how do you afford like how do you create that? So there's these growers themselves that are creating fantastic varieties, and people are finding lots of different ways to get into plant breeding from other angles, which is fantastic. That's how it's always been. But what are the those systems we want to create to be able to train more plant breeders to be able to write down what works and can keep access to all these great seeds. So I think that's on the boring side. There's a lot of behind the scenes stuff, but otherwise, I think on the fun side, there's so much going on with people starting to embrace flavor, the history, where something's from, who's done it, what's the background, and how to actually make it more than just a bin of like a generic crop, but actually reward people for seeking out new flavors. That I think will continue to thrive.
Awesome. Thank you so much for a fun and informative interview, Michael. This has been awesome.
It's great to talk to you. Thanks for the invite.
Thank you.